Hello, it's coffee time. If you want to see that, this is something that a student got me a couple of years ago. Pull it back there. There we go. Got polio, me neither. Thanks, science, or all these other diseases. Yeah. After a particularly rough virology class. Rough for me. Yeah. Fine for everybody else, I think. Yeah. All right. So, let's go. All right. So, there's the idea that viruses having a parasitic lifestyle are fundamentally selfish, in that the way they replicate is to go into a cell to use things from the cell until they are used up and then to move on to another cell. So viruses, they reap, they do not sow. And I don't even know that they reap, they just raid the cellar after you do all the work. Yeah. And yeah, like mice, but without the cuddly, fluffy, cute little face and beady eyes. Yeah. Yeah, it's the idea that I don't think it comes from, but it was popularized in popular science by Richard Dawkins. The idea of the selfish gene, and here's a cool little selfish gene. Uh, cartoon, apparently by Dawkins. Yeah, who knew? Yeah, appreciate that. Okay, yeah, and so there is the idea from Mart Krupovic and Eugene Koonin that viruses may be selfish replicators, just things that started out early, like at the dawn of time, and were able to copy themselves and never acquired and then lost some of the other metabolic genes that you would need in order to harvest the food and make a living and just do stuff in general, stay alive. And so that they've been parasites from the beginning, parasites once, parasites always, and always will be. And yeah, interesting idea. So the idea is that some of these replicons then would have got together and would actually be some of the things that helped to create the original cell, because cells do replicate. They do virus things and more. Yeah. And viruses do cell things but less, which is why they go into cells to raid the pantry and get all the things that they lack. Yeah. And so the idea would then be that viruses and cells have basically the same origin. And it's hard to even say that one comes before the other because they're both co-evolving the entire time. Interesting idea. Yeah. Let's look at coronaviruses specifically. So here we go. This is looking at coronaviruses, and you can see that these are all genes. You'll know some of these things. So NSP4 is going to be one of the genes that forms the pore, that the six-pointed pore that leads to the double membrane vesicles, where we think the coronavirus RNA may be manufactured. Then it comes out the pore and gets loaded into virions eventually, and everything is great. So this is just showing that every single piece of equipment, from this thing called the Y domain, and we think it's involved in the DMV pore. It may be the thing that is sort of controlling what gets into and comes out of the pore, like sort of the plug in the center of it. And that's one idea, but we don't know for sure as I'm recording this anyway. But from there all the way to the end of polyprotein 1B, which is the NSP16 methyltransferase, all those things are conserved, and they're conserved at frankly ridiculous E values. So the E value is the chance that you're wrong, the chance that this isn't the same gene. And yeah, every single gene is the same thing, comes from the same origin. Coronaviruses basically have one point of origin, and that point of origin almost certainly had all this stuff, because you find it in every single one of them. The only cases where we don't, which are gray here, are cases where we only know half the genome. And darn it if that half the genome doesn't look a whole lot like the ones that we know. So, there you go. What do these machines do? So, if you look at the wider picture, these are some nidoviruses. Nidoviruses is the order that's going to include the family Coronaviridae, which are all your coronaviruses. And so here's a coronavirus on the top. So we've got these little color-coded proteins. And the parts that started being conserved are starting with this brown part and ending with this gray part. So all of that. If you look at other things that are outside coronaviruses, but still within the order Nidovirales, and they do a lot of things similarly, what you'll find is that some of them have something like a spike, but quite often they do not. They'll have other things replacing that. There's usually something like the nucleoprotein, but those nucleoproteins are so distantly related to each other that you can't even be sure that they're actually the same thing. They might be. And a few very different seeming ones have been solved and actually make roughly the same shape. So it is quite possible that they are all homologous, like sharing a common ancestor. And you can see that everything in the front part of the genome, all these sort of white squares and all these colors that show up a couple of times and then never again, these are areas that are highly customized in the genome. And so these are areas where you tend to not get conserved things. So the stuff that makes a coronavirus is up here. The stuff that makes it dangerous is going to be up here, because these are going to be things that are specific to that virus, and they almost always involve interacting with some part of their host and disabling, dismantling, modifying some part of the host behavior in a way that, darned if it doesn't always seem to benefit the virus. All right. Nidovirales are part of a group that includes things like picornaviruses. So these are the Pisoni vircites, and they're all going to have a helicase of some sort, although it's a different helicase in this group, and a protease, a main protease, and a polymerase. So here's the polymerase family tree. And you can see the branches on this family tree have been color-coded based on how much evolution they've done. So the length of the branch is the total, evolution in terms of nucleotide changes. The color on the branch is how many times along the way they have stopped, picked up an entirely other gene from some other source, and we never know the source for sure, and incorporated it into their genome, or taken something like an entire gene from their genome and just ejected it, and they've been fine. Yeah. And so you can see, compared to some of these other groups like Picornavirales, they have nice long branches. They've been around for a long time, but they are not good at pivoting. These things have mostly black and blue, which are the colors for the least amount of big changes, like adding and subtracting entire biological machines. Look at the nidovirus branches. You can kind of see it over here, and we zoomed in on one part. Look at that. Those are some pretty bold, bright colors, because nidoviruses are specialists at picking up and discarding whole domains. That's the kind of evolution that makes a virus unpredictable, and I think that's the kind of evolution that makes nidoviruses less predictable. So coronaviruses are for sure in this group, and it does kind of make sense, doesn't it? Yeah. These are viruses to watch, because these are viruses that change in ways that are hard to predict. So, from the study of sort of viruses as a group, we can learn these things that at least inform us on what's going on and why bad things happen to good people and also bad people, because viruses don't care. Yeah. And so here's that shown as a graph. This is how much change a picornavirus is able to make. A little tiny bit of change. Look at them. They're doing their best. Oh my goodness. Yeah. A little round of applause. Participation badge for the picornaviruses. And then you got two groups of viruses that are on the way to becoming nidoviruses. They actually have the helicase in common with nidoviruses, and they are much closer to nidoviruses. So they're like the steps before you get to these. And they actually have an intermediate amount of domain level change, and then you've got your actual nidoviruses, and they are off the charts. They are ridiculously overpowered when it comes to adding and subtracting whole big biological bits of themselves. Yeah, and so, I don't know, these are the things that they interest me and also maybe worry me. Yeah, this is why you don't let nidoviruses run loose, because they don't ever stay the same, and they change more and more dramatically than anything else that I know. Yeah. All right, so there ends section 2. We will get into this with section 3 in just a moment, or tomorrow if you've had enough of virology for tonight. Anyway, see you in the next one.